Hello, so I'm back again to talk about glycolysis and the citric acid cycle. So a little bit of an overview. Glycolysis is the process responsible for breaking down glucose into an even simpler molecule called pyruvate. Glucose is a basic form of carbohydrate called a simple sugar and one of the most common sources of energy. So our bodies ingest food, and then the food from that carbohydrates are extracted, and that's how we get glucose, and then glycolysis occurs. During glycolysis, one glucose molecule will turn into two pyruvate molecules. Glycolysis has two phases. In phase 1, two molecules of ATP are consumed by the cycle to generate the energy needed to break down the glucose into another molecule. This molecule is converted into the pyruvate. Breaking apart the molecules releases the energy within the molecular bonds, and that's what is packaged as the ATP. The second phase will yield the 4 ATP, but since 2 ATP were used, 2 more is gained. Glycolysis yields 2 molecules of NADH, which is a molecule important to pathways as an electron donor. Once glycolysis ends, the citric acid cycle occurs. This cycle is supposed to release the energy created in the pyruvate molecules during glycolysis. The citric acid cycle occurs in the mitochondria of the cell and will eventually break pyruvate all the way down into organic substances like carbon dioxide and water, thus releasing all of the energy inside the molecule. So in step 1 of this process, acetyl-CoA is generated either by oxidative decarboxylation of pyruvate from glycolysis, which occurs in the mitochondrial matrix by oxidation of long-chain fatty acids. Acetyl-CoA then enters in the TCA cycle, where it is oxidized for energy production. Acetyl-CoA acts as starting point as well as a fuel for the citric acid cycle. Acetyl-CoA is produced from pyruvate through a series of reactions. As we already know, pyruvate is the endpoint of the glycolysis pathway, and under aerobic conditions, pyruvate is transported to mitochondria by pyruvate antiporter. Pyruvate that is derived from glucose and other sugars by glycolysis is oxidized with the help of pyruvate dehydrogenase complex, a cluster of the 3 enzymes. Sorry, big words. Glycolysis produces the molecules that are processed by the citric acid cycle. The citric acid cycle occurs in the mitochondria of the cell, as I just mentioned, and will eventually break the pyruvate all the way down into the inorganic substances. Pyruvate is produced in the cytosol and transferred to the mitochondria. Oxidative decarboxylation of the pyruvate takes place with the help of the pyruvate dehydrogenase enzyme, a molecule called TPP from temporary bond with pyruvate, as you can see in this photo. And then in step 2, the enzyme dihydrolipol transacetylase helps to attach other molecule lipoamide, and TPP from the first step is removed to form the acetyl group. For steps 3 and 4, the CoA group attacks the acetyl group and forms acetyl-CoA and lipoamide released in reduced form. As you can see in this photo, this is an overview of the lipoamide being transformed, and then it is further reduced. And then in the final step, the lipoamide is reoxidized with the help of the dihydrolipol dehydrogenase and also NAD, which becomes NADH, while the oxidizing lipoamide. So that is the next cycle. For the next cycle, the lipoamide can be used. So that is how that process works, and thank you for listening.